In this unit, we'll talk about colligative properties, what they are, and to what extent they can affect a solution. Colligative properties are properties that depend only on how much of a material is dissolved in solution and not what is dissolved in the solution. Colligative properties include vapor pressure, boiling point, freezing point, and osmotic pressure. First, with vapor pressure. Raoult's law describes vapor pressure as that the pressure in solution is equal to the pressure of the solvent in its pure form times its mole fraction. That is, mole fraction is the moles of a component divided by the total moles present. Boiling points are elevated by introducing materials into solutions. Again, it doesn't matter what the material is, simply how much. Add more material to a liquid, the boiling point will go up. Freezing point is like boiling point, except rather than increasing the temperature at which something boils, freezing point decreases the temperature at which something freezes. It's one of the reasons why we add salt to roads when there's ice. The salt ends up lowering the freezing point of the ice so that it melts and runs off the road. We can quantify the change in boiling point or freezing point by taking a constant known as the Van 't Hoff factor, multiplying it by a boiling point or freezing point constant, multiplying it by the molality, in this case how many moles per kilogram there are. The Van 't Hoff factor is a fudge factor because based off of how many particles and not what the particle is, the Van 't Hoff factor seeks to correct for materials which are ionic. A covalent molecule, when you place it into water, stays as one molecule. But when you put an ionic compound into water, it essentially breaks up into 2, 3, 4, or 5 different particles, depending upon what the subscripts are on the ions. So something like sodium chloride, the Van 't Hoff factor would be 2 because it's 1 sodium and 1 chloride ion. Potassium sulfate, 2 potassium ions and the polyatomic ion sulfates for a grand total of 3. Osmotic pressure is also a colligative property and one which we tend to pay a little more attention to in biology and nursing. Osmotic pressure is the pressure required to take a fluid at equilibrium and push it back so that it has an equal fluid level on both sides. That is how much force is required to push down on our middle tube so that it restores to the tube on the right. That's the osmotic pressure. Osmotic pressure is equal to our Van 't Hoff factor again, times molarity, or number of moles divided by volume, times the gas constant R, times the temperature T, and remember T must be in Kelvin. Osmotic pressure can be used to describe solutions when there's a separation or barrier such as a blood cell. We have isotonic solutions where the osmotic pressure is essentially the same on both sides of the cell or a membrane, and so there is no change in water levels. And this is an ideal situation because when we introduce fluids into the body, whether it's a saline solution or a blood transfusion, we strive to have isotonic solutions because the last thing we want to do is have our blood cells explode or implode. A hypotonic solution has a lower osmotic pressure, so solvent will leave the solution at a higher rate at which it enters. And so what a hypotonic solution causes is a blood cell to implode if we're not careful. A hypertonic solution is just the opposite. We have a high osmotic pressure, and solvent will enter the solution at a higher rate in which it leaves. When we describe this in terms of biological function or how a cell reacts to its environment, we describe something like a red blood cell as having a cell wall, which is a semi-permeable membrane. That is, it's a selective barrier of what it lets into and out of the cell. If a cell is in a hypertonic solution, then we undergo a process known as crenation, where the blood cell shrinks up. And if we're not careful, we'll actually explode or implode. On the other hand, we have hemolysis, which is just the opposite. We have a bunch of water running into the blood cell, and so the blood cell will explode. So these are things you have to be careful of, and you'll probably see again if you go into a nursing program.